To understand this lecture properly, you need to understand the concept of the isoelectric point again, okay, if you have not done it already. And the second thing you need to do is how amino acids they change their charge. Okay, so how amino acids vary their charge as a function of the pH. Okay, so these two things you must know before you understand this lecture. Okay, so if you don't remember, I am advising you to go back and understand it first. Okay. So, to start with, let's understand the concept of the protein primary, secondary, tertiary, and quaternary structure. So, the simplest level of protein structure is the primary structure. So, it is simply the sequence of amino acids in the polypeptide chain. So, this is the structure of the hormone insulin, and which has two polypeptide chains. So, this is A and B, and shown in the diagram. So, this also includes the disulfide linkages, and this can be so any chain. If you are writing the sequence, for example, if you are writing the sequence of the A chain, okay, glycine, isoleucine, valine, and all the way to asparagine. So, as I discussed earlier, this always starts with the N terminal, and this always ends with the C terminal. Okay, so that is um, without any exception. Similarly, this would start with the N terminal on this side from the writing perspective, and this should end with the C terminal. Okay, so that is how amino acid structures primary structure is written, or protein primary structure is written. So if it's written in a single letter code, for example, if you're writing this as G, okay, K means lysine, R means arginine, and so on. The N terminal, the left side is always the NH2 and the right side is always the COH. So this is true for primary structure. The second kind of structure is a secondary structure. Secondary structure is the existence of helix and the sheets and turns. Okay, so for example, if you have a particular protein sequence, it tells you that this part of the protein sequence will become a helix, this part will become a sheet, and this part will become a, turn, for example. So for example, this just giving a random example here, for example, the A chain here, let's say if you can build you can do a theoretical prediction that this part will build a helix, alpha helix, this part will become a beta sheet, okay, and this part will become a helix again, and this part will become a turn. Okay, so this information is part of the secondary structure information. What it does not tell you is that, so this helix, which starts with a helix, then a sheet, which is like this, okay, followed by a third helix, and then a turn. Okay, so how all these elements form the total protein in a globular shape or in a fiber shape or any other 3D shape. So that information is not part of the secondary structural information. So this is secondary structure, and this is does not tell you anything about the any further about the 3D arrangement of them of these elements in the protein. Okay, so that is the restriction of the secondary structure. So as I said, this includes the alpha helix, beta sheets, and so on. So we'll learn more about this when we learn how to predict the which one will form alpha helix and so on. The third structural information is a tertiary structure. So tertiary structure is now how this helix and sheets and turns they form. So let's say there is a helix right after the other, then there is a sheet. How they form together and form this kind of 3D structure. Okay, so that is the tertiary structure. So the overall three-dimensional structure is the tertiary structures. Okay, so how it forms, so basically we'll learn more about this. The hydrophobic groups they usually bring together, and the hydrophilic groups they usually stick outside and interact with the water. So interior of the molecule they have the hydrophobic groups interacting with each other, and the hydrophilic groups they stay outside and interact with the water. Okay, so that's how tertiary structure is formed. Also, at this stage the disulfide linkages are formed, and we have already discussed this and shown this using PyMol, that disulfide bonds are a very crucial component of forming the tertiary structure. The final one would be quaternary structures. So quaternary structures are sometimes, what it discusses is that sometimes proteins are made up of single polypeptide chains, and so this would form only one subunit. Okay, and this one unit is the entire functional protein. However, sometimes more than one subunit is required to make a bigger actual functional protein. So for example, this is one subunit. This requires another subunit to form a complete structural functional protein. So not that it could be covalently linkage, it could have a disulfide linkage or may not be, and simply sometimes there's an electrostatic interaction. But once they form together, they actually become a functional protein. Okay, so this structure together is the quaternary structure. Okay, so these are the four different kind of structures or the coordinates. So here are some examples. So GAPDH, it is made up of 4 subunits. DNA polymerase is made up of 10 subunits and so on. Okay, so and so on. So this is a summary slide showing the all 4 types of structure. This is primary structure, we just have the sequence and nothing else. And when we write them in the sequential form, like in the linear form, usually the left one starts with the NH2 and the right one, whatever it is, ends with a carboxyl, so the C terminal. Okay, the secondary protein structure is how the structures from sheets and helices, but that's all. It doesn't tell you how they after that they interact with each other. Okay, so it can tell you that so this part of the structure is a helix, this part of the structure is a sheet, and so on. Okay, that's all it can tell. So after that, the tertiary structure is how the structures folds to form a tertiary structure. Okay, and quaternary structure is when, let's say this is this particular one, and it interacts with the second. This then it will be called a subunit. Okay, and then it will be interacting with another subunit. This is subunit 2, and then they will form together to form a quaternary structure. So some proteins, as I discussed, will have only a tertiary structure. They will not have a quaternary structure. Okay, some of them will do. Some of them will require to form quaternary structure to form functional unit. Okay, so that is a difference. Okay, so that is the difference between quaternary and the tertiary structures. So next, what we are going to discuss is about proteins. Is that so? Proteins do come in various shapes and sizes. Okay, and you can see that the molecular weight of the protein can vary from as something as small as 13,000 to as something as large as 200 2900,000 and so on. Okay, this is the number of residues. So one key point is that so all of this you can see that mostly they have one polypeptide chain. So this particular one, the big one, has a one continuous polypeptide chain. Okay, interesting, isn't it? So the average molecular weight of the proteins is 110, meaning if you divide this by this will be average 110. So, or in another way, if you are given the number of residues, okay, and if you multiply them with 110, you are supposed to approximately get the total molecular weight of the protein. Okay. Second point is how the molecular weight is represented. So molecular weight, we know that it is represented by gram per mole already. Okay. So, but usually that is not the term we use it. So this is what term we use is called Dalton. So Dalton is the same term as the gram per mole, and but we use for proteins we use the term Dalton to represent the molecular weight. But remember, this is the same term as the gram per mole. So next few slides or next few classes, what we will be discussing is that the protein chemical properties and various analytical techniques and methods for separation of proteins from crude mixture and all. So what this will talk about is that, let's say you have prepared a protein either chemically or you have prepared it by expression, meaning in a cell, in a bacterial cell or in a mammalian cell, you have prepared your protein in large quantity over other native proteins. Okay, So there are other proteins above which a protein is prepared, or you can also prepare this chemically. uh, we'll discuss both. So what you want to do, you how do you purify this and isolate your protein of interest? Okay, so we'll learn those. That will be called the separation techniques, which will be used to separate the proteins from other unwanted proteins. So there are different types of protein separation techniques, and we'll learn all of them, or at least some of them. So you can use the protein's properties, such as chemical properties, such as side chain properties, the hydrophobicity, hydrophilicity, size, charge, all this to separate a protein of your interest. Okay. So again, what we are going to learn, this is a column chromatography. Okay. So we'll do a short lab on the column chromatography in the next lab. Okay. So the first one we are going to discuss is the ion exchange chromatography for protein purification. So ion exchange, as the name suggests, it relies on the charge of the protein. Okay, so how does this protein's charge come into the picture? Okay, so these are for, before I discuss that, so these are some links that you can go over. This particular one, if you are thinking from an experimentalist perspective, this one from a theoretical understanding, and this is for a short overview understanding. Okay. So, so this picture gives you a broad overview of how this process works. And before we go ahead into detail, so in this column, what we have, this is polymer beads with negatively charged functional groups. Okay, in this you are adding proteins where they have different types of positive charge. Okay, so you can see blue has net negative charge, dark blue has large net negative charge, light red has net positive charge, and dark red has large net positive charge. So once you put it, basically this two which has negative charge, they will be repelled by the groups and they will quickly come out and be collected as a eluent. Okay, they will not interact with the beads, the functional polymer beads, but your positively charged groups, they will get stuck because of the ionic interaction or electrostatic interaction. So how do you get elute them then? You what you do, you flush the column with positively charged ions, okay, Na plus, imidazole, or some other ions, which then, if you supply them in a large quantity, they will bind the beads. Okay, so the beads are negative, they will bind the beads and let the protein go. Okay, so the proteins then will be collected in the column, in the as a form of the eluent, not in the column but as a form of the eluent. So eluent means something that is coming out of the column. Okay, so that is how your proteins will be separated by charge. Okay, so this is the principle, so we'll go over it over the more details in the next few classes. Okay, so that is all.